Hi guys, today I'm going to be talking about my role model. Dr. Marcela Uliano da Saliva grew up in Brazil and fell in love with nature, specifically rivers, at a young age, inspiring her to study biology. Dr. Marcela studied biology at the Federal University of Rio de Janeiro, specializing in the study of computational biology and genomics. Dr. Marcela also received her PhD in biophysics from the Federal University of Rio de Janeiro. Dr. Marcel's biggest obstacle was getting funding in Brazil for her research in ecology, along with protecting the ecosystem while genetically modifying an invasive species. Dr. Marcela has expressed much love for the outdoors, specifically rivers, and observing animals in their natural habitat. Dr. Marcela has also expressed much love for her Brazilian culture. Dr. Marcela inspires me because of her drive to solve a real world problem she was passionate about since she was a little kid. Dr. Marcela is genetically modifying invasive species, which connects to me because she is following her passion for science that was established when she was a kid. Dr. Marcela works in molecular and genetic biology, synthetic biology, and even though Dr. Marcela isn't trained in ecology, she works on environmental conservation by trying to genetically modify invasive species. Questions Dr. Marcela is working to answer are how you can genetically modify invasive species to not be able to reproduce without harming the ecosystem. What specific genes make invasive species so successful at reproduction in a short period of time? How can we create a species-specific genetic tool that can only target this invasive species? The Linopron fortinu mussel, also known as the golden mussel, is an invasive species in the South American freshwaters. Dr. Marcela and a group of scientists worked to establish its entire genome. 3 golden mussel individual species DNA was extracted from their gills using a D-Nessie blood and tissue kit. A collection of their DNA was sequenced from a machine called a high-sequence 1500 machine. A machine called a P4C6 sequenced the chemistry into their cells. For translation, 4 tissues were sampled for the RNA, which was extracted using a NextFlex rapid directional RNA sequence kit. This extraction was read from a NextSequence machine. The sequences that were collected were put into a software by the company Pacific Bioscience that was ran by a program called RNA Scaffolder, which uses the amount of distance between each exon in a genome sequence to connect the scaffolds. They then proceeded to use a machine to fragment the DNA, which came back to be N50 in length and not higher than 5 kb. All of this information showed that 81% of the golden mussel was orthologs. This data table depicts the taken to assembly the golden mussel genome. The genome begins depiction by small fragments DNA that connects together, known as contig. Next, they will use those contigs to build scaffolds. They will use technology DBJ20LC, correcting any errors and creating longer scaffolds. Transcriptome scaffolding is the third step, joining scaffolds together with RNA sequencing. The final step is RNA and DNA resequencing, causing them to be realigned and sequenced. The assembly of the genome for the golden mussel was Dr. Marcela's biggest uncovering so far. In the future, Dr. Marcela hopes to identify certain genes that are helping the golden mussel be so high successfully at reproducing. Dr. Marcela also looks to find targeting mechanisms to inhibit these genes from being transcribed. In conclusion, Dr. Marcela is making great progress on working to use genome editing to prevent invasive species from over-reproducing and bringing harm to the ecosystem. Dr. Marcela has used her love for nature as a kid to inspire her to go into this field of research. Dr. Marcela is a real-life inspiration for many and is making a difference every single day in the world.